Beyond transcriptional activation, which post-translational modifications, such as phosphorylation or methylation, for instance, seem most pivotal in mediating inflammatory resistance mechanisms?
I think phosphorylation is obviously the first because the activation of inflammatory signaling requires specific protein phosphorylation. So as I mentioned earlier, JAK1, JAK2, STAT1, STAT3, STAT5, they require phosphorylation, autophosphorylation for the alteration of their activity and transcriptional modification eventually by STAT, for example, which is a key for inflammatory signaling. Now, obviously, the protein levels and activity is affected by other post-translational modifications such as ubiquitination, which has been found to change significantly. One other thing that's very interesting is how to link spliceosome changes in the splicing of the RNA with activation of inflammatory signaling. We know that altered splicing of specific genes, which is caused by alterations of the spliceosome machinery, which are seen very much in MDS, for example. I remind you, U2AF1, ZRSF2 or SRSF2, and other spliceosome molecules are altered in MDS and AML, and that leads to alternate splicing, which affects significantly a number of components of nuclear factor kappa B and TNF alpha, for example.
Mhm. Have you observed specific modifications or signaling proteins or metabolic regulators that could serve as actionable nodes for drug targeting?
And you are referring to both inflammatory pathways and metabolic
Yes.
alterations. So more recently, we know that metabolic alterations are critical in a number of. I mean, we obviously know that AML with IDH1, IDH2 mutations is critical. There are critical changes in metabolic signaling. However, we know that TP53 mutated MDS AML also has characteristic metabolic abnormalities. Not targetable at this point, but I would hope that we will
Mhm.
understand better how to target these pathways and then can introduce a number of new drugs for this. So metabolic alterations for sure. Inflammatory signaling, I mentioned most of the ones that are promising, such as nuclear factor kappa B, I think TNF alpha signaling, but also JAK-STAT. And I will insist that potentially STAT inhibition has not been studied as well as it should be in leukemias.
Mhm. These post translational events differ between primary resistance and acquired resistance after therapy exposure?
So post-translational modifications that are different between acquired resistance and primary refractory disease, yes, we know that primary refractory disease and acquired resistance sometimes are different. We would expect post-translational modifications to be a little more prominent in acquired resistance in most of the cases, but we do see some of those changes even in primary refractory disease. I think the main factor that alters those post-translational modifications is the initial mutations or the acquired mutations. So we shouldn't forget that all these events are associated with specific somatic mutations in most of the time, and they differ very much between different subgroups of leukemias.
Mhm.